Hello, everyone. It's my pleasure to welcome you to today's webinar by the Global Antibiotic Research and Development Partnership, GARDP. My name is Shireen, and I'm going to be your host today. So just a quick word about Revive, which is GARDP's education and outreach program. It aims to connect and support antimicrobial research and development community by facilitating learning, connecting people together, and sharing knowledge. So the webinar of today is part of our education activities. You can find all of our webinars that are recorded on the as the webinars. We also have other activities on the Revive website. So we have our series of antimicrobial viewpoints, which are articles where experts discuss various topics within their fields. We also have a resources section that includes the antimicrobial encyclopedia. So this has definitions on various important terms that you will hear, for example, in today's webinar. And some of these terms include video where an expert will develop the term further. As always, today's presentation is followed by a question and answer section. So just a reminder to our audience, you can submit your questions throughout the presentations by using this functionality on the platform. And if your question is directed to someone specific, you can please include their name on the question as well. So we'll try to address as many questions as we can at the end of the three presentations. Today's speakers are Mark Blaskovich, Matthew Todd, and Luiza Galarion. And our moderator today is Heike Brötz-Oesterhelt. Heike is a microbiologist, and she has worked in antibacterial drug discovery in academia and pharmaceutical industry settings for over 25 years. After earning a diploma in biology and a PhD in microbiology, she joined the anti-infective department at Bayer Healthcare and later co-founded the anti-infective biotech company A-Curis. In 2010, she returned to academia as a professor of pharmaceutical biology at the University of Düsseldorf. Since 2014, she has been a professor of microbiology at the University of Tübingen at the Department of Microbial Bioactive Compounds. Heike is co-speaker of the Cluster of Excellence Controlling Microbes to Fight Infections and deputy speaker of the partner site Tübingen within the German Center of Infectious Research, DZIF. She is particularly interested in unraveling the molecular mechanism of new antibiotic agents and the operation modes of novel antibiotic targets. Welcome, Heike. I'm happy to hand over the floor to you.
Yes, thank you very much, Shireen, for this very nice introduction. And it is my pleasure to host today's seminar. Yes. So today's seminar is on data-driven antibiotic discovery. And yeah, data-driven science, what does that mean? In a literal sense, this term would mean what all of us do, right? We generate data, we analyze data to gain scientific insight. But the way the term is commonly used also includes the compilation and use of large data sets and also data sharing. And it is in this sense that I want to discuss a few points. Yes, why it is essential to share data and make them openly accessible according to the fair data principle, which means that data should be findable, accessible, interoperable, and also reusable. Yes, it is important because bacteria are tough opponents. They are really challenging to overcome, which is our goal, our joint goal, especially nowadays with soaring resistance rates. And we need all the help we can get, and community efforts are powerful. we race against the clock. Yes, you all know that antibiotic resistance is soaring. So if we want to avoid a post-antibiotic era, we have to minimize the time for our learning cycle. And most of the current antibiotic discovery and development is performed in small to medium enterprises, small companies, and at university, as most of the big pharma companies have stopped their efforts in antibiotic discovery. And in this environment, it means you have PhD students coming and going, finishing after their thesis time. You have small companies that have only funding for a limited time frame. So we have more rapid turnover also of scientists, and we need to preserve knowledge and work along also regionally dispersed pipelines. So there is some group that has this particular assay and capacity, and the compound needs to be further profiled in another place on this continent, a continent, or even globally. So, and most of current antibiotic research is financed by public funds, and we should try to spend taxpayers' money efficiently and wisely. So today's seminar is about data sharing, and we go from a gap analysis to also solutions. And we have come a long way already, but it is also clear that there is still room for improvement. And this slide is like a kind of pep talk to motivate all of you to join in, because each analysis that we do provides us with new insights into how we can do better and how we can overcome bottlenecks, and we will get continuously wiser. And with each joint and successful community effort, we will see how motivating this is, and this will inspire new collaborative efforts, and we will keep getting faster. And we do not have to carry all the responsibility and workload alone. Yes, we can pick our community brain, and it is much more fun to work together with colleagues. Antibiotic resistance is a global problem. Good antibiotic agents are a global, joint, and also, unfortunately, limited resource. And yes, let's make antibiotic discovery and development also our global task. So with that introductory comments, I would like to introduce our first speaker of today. So we will have Mark Blaskovich. Mark is Director of Translation for the Institute of Molecular Biosciences at the University of Queensland in Australia. Hello, Mark. Nice to meet you. We know each other from conferences. Mark describes himself as an antibiotic hunter. Yes, he's always out for the next interesting antimicrobial agent. And in that sense, he also leads the COAT initiative, which is a huge crowdsourcing effort in which he and his team collected molecules from chemists all over the globe. Yes, I think, Mark, it is the broadest and widest chemical space that has ever been mined for antibiotic activity. He also leads the Center of Environmental and Agricultural Solutions to antimicrobial resistance, which is also interesting from a One Health perspective, because we all know that discovery of new agents alone will not solve the AMR problem. We also have to preserve the ones we have and the new ones that we will find in the future. Mark has also a strong background in industrial drug discovery with multiple ongoing collaborations, and he also serves on the Revive review panel. And today, Mark will speak about the challenges in antibiotic discovery, our learnings from the past, and concepts and ideas how to do better in the future. The floor is yours, Mark.
Thank you very much, Heike. Let's wait for my slides to appear. Great. So I'd like to thank Revive and GARPI for the invite to present. I've been tasked to talk about the scientific bottlenecks in antibiotic discovery and how data sharing collaboration can help address some of the issues with the bottlenecks. So this is very much a personal viewpoint of what the bottlenecks are from my experience. There definitely are others that I won't be talking about, but hopefully I'll give an overview of what I think are some of the most significant challenges. So I thought, not knowing what the diversity of the audience was, so bit too far. Go back one slide. Given that not knowing the knowledge of the audience and the background, I thought it'd be really good just to set the stage initially and highlight what one of the major issues facing us because of antimicrobial resistance is and how antimicrobial resistance is becoming a problem. So antimicrobial resistance is nothing new. Every single antibiotic that's been introduced, starting from penicillin in the 1940s, resistance has been observed even before the antibiotic has been used clinically in many cases. So if resistance is nothing new, why has it become such a problem now? And it's because the discovery of new antibiotics is not keeping pace with that development of resistance. So from the 40s to the 80s was the golden age of antibiotic discovery, when we were discovering completely novel classes of antibiotics acting by different mechanisms. But since about 1980, we've really not discovered any new classes, or one. And so this fundamental lack of development of new antibiotics means that as the bacteria become resistant, we don't have new tools to be able to treat the infections caused by those drug-resistant bacteria. Reflected in another viewpoint, the number of new antibiotic approvals since 1985. So you can see again, even in the 90s, there were significant new antibiotics being developed, which were new generations of existing classes. But even that development has declined over the years. And so we're just not fundamentally developing new antibiotics fast enough anymore. So highlighting bottleneck number one is a loss of knowledge. And the reason for that is because of the decline in antibiotics, almost every big pharma company has exited from fundamental antibiotic research since that golden age of antibiotic discovery. And so we've gone from tens of thousands of researchers in industry focused on discovering new antibiotics to basically only a handful. And this has been tracked in a report that came out a year ago called Leaving the Lab, which tracked the decline in the researchers involved in antimicrobial resistance. So looking at the number of publications has been declining for over 20 years versus the number of publications in cancer or HIV/AIDS, which are still on a steep increase. And that's accompanied by people exiting antimicrobial research. And so they tracked a number of the career paths of people who have left companies that focused on antimicrobial resistance when the companies have either exited antimicrobial drug discovery or they've gone bankrupt. And very quickly, the first generation, less than a quarter of the researchers stayed in the antimicrobial field. And then the second generation, another third decline. So you're having this loss of knowledge of people that have the practical experience of how do you develop new antibiotics. And so estimates are anywhere from approximately 500 to 3,000 researchers globally that are still focused on trying to discover, develop new antibiotics. And so with that is that this loss of cumulative knowledge from all the people that were working in pharma on the intricacies and specialties focus needed for antibiotic development. And so one of the needs for data warehouses or sources where people can go for information is trying to retain all this collective knowledge. So bottleneck 2 comes down to a lack of new chemical diversity. So antibiotics are not drug-like. They don't obey a lot of the traditional medicinal chemistry rules, in particular rule of 5, which is trying to predict characteristics of compounds that will make them orally available and desirable drugs. When the rule of 5 was formulated, it explicitly excluded natural products, and almost all antibiotics are derived from natural products, so it explains why they generally don't comply. They often don't look drug-like. So they have these reactive functional groups that in any current medicinal chemistry screening program would be immediately removed from further consideration because of the potential for toxicity and side effects. And so antibiotics have all these properties where if you're applying modern medicinal chemistry techniques, you're going to be missing many of the antibiotics that were previously discovered. And so if you look at a typical corporate screening library now, in terms of molecular diversity, just looking at lipophilicity and molecular size, they're very narrowly focused now, constrained by this rule of 5 parameter. If you go to a publicly accessible database of published academic compounds, the Campbell database, you can see that there's a huge amount of chemical diversity of compounds, different structural diversity of compounds that do have antimicrobial activity. And so if you're only searching for chemical diversity in this small space, you're missing out on all this other chemical diversity that has a larger proportion of compounds that might have antimicrobial activity. And so there's a lack of novelty in the current collections of compound libraries. So, big pharma's screened all their large collections. The compound collections you can buy from companies such as ChemDiv are all very constrained in terms of the overall chemical diversity that are contained within them. And because of that, you're relying basically on everyone around the world is relying on the same compound screening libraries from the same commercial vendors. So there's no new novelty to be found within those libraries. And in terms of chemical diversity, until recently, natural product discovery, which was sort of the source of all original antibiotics, has generally focused on culturing the same species under the same conditions, and so you end up just rediscovering the same antibiotics. And so there have been a lot of advances over the last decade or so applying new technologies trying to supercharge natural product discovery, and there have been a lot of new discoveries that are now winning their way through of new classes of antibiotics because of these metagenomic sequencing, forcing compounds under different culture conditions, etc. And then the use of generative AI is now very much a hot topic, but unless it's constrained properly, it can design wonderful molecules that are just impossible to synthesize. And sorry, just go back if I can to that one. And so there's a recent article just came out this month in PNAS looking at generative AI trying to navigate synthesizable chemical space rather than just making any molecule it can think of. Bottleneck number 3, if you're looking at standardized large data sets. And so the application of artificial intelligence now, it's like the old computer systems. If you put garbage in, you get garbage out. And the biggest problem with AI is it really needs large standardized data sets in order to be able to do accurate predictions. And so if you look at the most successful AI application, which is really AlphaFold, that's reliant on the protein data bank, which has, as of 2023, it had 200,000 structures that have been inputted over the last 30 years. And so there's this huge, carefully curated data set that AlphaFold relies on, which is not available for most other applications of artificial intelligence analysis. So model building requires very clean data. So where do you find that clean data, and how do you do it? So you can try looking at Campbell as a source of compounds that have antimicrobial activity. So there's almost 2 million compounds listed in Campbell. Within there, about 250,000 of those compounds have antimicrobial data. If you're focused on antimicrobial MIC data, now that's reduced greatly down to only about 77,000 compounds. If you now try looking at that data for a single organism, so that data is spread across multiple organisms, so only about 1/4 of that data is with E. coli. But for standardized data, you want to be using a specific E. coli strain. And so more than a third of the strains, third of the data doesn't actually specify what strain. One standardized strain, ATCC 25922, makes about 30% of the data, so that's the most common one, but now you've reduced your data set by another 30%. And then you look at assay conditions. So you've got broth dilution assay, you've got agar disc diffusion assay, or you've got undefined assay conditions, which means you can't really compare the data. So now you've cut your data in half again. And so from all that large data set you have, you come down to 8,000 compounds that have broth microdilution data against a single E. coli strain. And so that's the level of the need for developing large standardized data sets, because the existing data sets trying to mine data, it's very, very difficult to build up large sets of data from what's being published. The other caveat with that is that the negative data, the compounds that don't have any activity, are often never reported. Bottleneck number 4, data integration. How do you tie everything together? And so in terms of antibiotic drug discovery, you want to be able to tie together the biological activity. And so you've got your antimicrobial data, IC50, EC50 of isolated targets, or percentage inhibition, or MIC of the organisms. You want to get further into the in vivo efficacy data. So you need standardized results, standardized assay conditions. You want non-actives, and how do you then handle combinations and synergies? Obviously, biological activity. There's also the ADMET considerations of in vivo, in vitro toxicity, pharmacokinetics, and again, how do you develop your standardized results and units and assay conditions? From a chemical structure perspective, how do you represent your data? Coming up with better standardized representations of protonation form of structures, of different tautomers, of different salt forms is very difficult to capture. For trying to integrate whole genome sequencing, Illumina versus long reads through ONT type of methodology, what type of program to use to assemble it. So again, all these variations in how you collect and interpret the data. And finally, trying to put all that into AI models. So can you use them to predict your antimicrobial activity? Can you use it for generative structure design? From a resistance perspective, can you go from genotype to phenotype? And so having a centralized warehouse we can try to tie all this data together would be very, very useful. And finally, there's collaboration. So if you go back to how antibiotics were discovered originally, they were all very collaborative efforts. So one example, vancomycin, isolated by Cornfield at Eli Lilly. He was sent soil samples collected in Borneo by a friend from the war, William Conley. And similar stories for the discovery of other antibiotics. People shared information, shared stuff around without MTAs and CDAs. And so, you know, in the current climate, we're buried under paperwork of having to get all these agreements into place before we can even talk to one another. So how do we enable the better transparency and collaboration across all these entities? Multiple small data sets held by individual entities, and a lot of entities trying to connect AMR researchers out there. There's not really one central go-to source. And so one example within Australia, the Australian Antimicrobial Resistance Network has been set up to help foster collaboration between academics and industry researchers to provide a focal point, but then that's replicated or potentially duplicated in other countries as well. And from a government perspective, there's a lack of single point government coordination. So again, specifically within Australia, antimicrobial resistance covers multiple departments, and even within one department, that's the most obvious one, Department of Health, there's separate areas focused on hospitals, therapeutic goods administration, reimbursement of antibiotic fees, the new Australian Centre for Disease Control, the funding agencies. And so how do you tie all this together and have a common source? So in the last few minutes I've got, I'm just going to really quickly go over a potential solution to some of these issues that we've been working on for the past 10 years. So back in 2015, we set up the Community for Open Antimicrobial Drug Discovery with the idea of trying to leverage collaboration by having a crowdsourcing initiative, trying to discover new antibiotics by untapping a wider source of chemical diversity. And so the idea is we had funding from the Wellcome Trust and the University of Queensland to set this up. The idea was to offer free testing to researchers around the world to send in their unusual chemical structures for antibiotic testing. And it's been very successful. We've tested over, by now, 350,000 compounds from over 350 research groups in 50 countries around the world. So there's a huge amount of interest in participating in an initiative like this. We've identified over 10,000 initial hits in our primary screen, which has done a single concentration against 5 bacteria, 2 fungi. And that's led to about 3,000 confirmed hits that have some selectivity over mammalian cell cytotoxicity. And so, this data has helped the collaborators publish articles, write grant applications, put in patent applications. So there's been some real world impact of what this data has been used for. In terms of trying to get over that barrier for collaboration of MTAs and CDAs, we've made it as simple as possible to submit a compound where we don't, from our side, need an actual contract signed or anything signed. We're using a software license type terms and conditions button you can just click on. If universities that are sending compounds still require a formal MTA, we've got a very simplified 2-page version, which seems to be pretty amenable for people. So in terms of is COAD achieving what we set out to do? Do we actually identify greater chemical diversity? So if you compare the chemical diversity of the COAD library in blue with the chemical library in a PCA plot in orange, we get very similar coverage. So we actually are tapping into that high chemical diversity that the academic compounds exhibit. If you compare it to that commercial library, we're much more diverse in commercial libraries, so indeed we are seeing greater chemical diversity. If you compare it to natural products, it's not quite as good. We've got some points that cover that natural product space, but in the totality, it's still more focused on structures that are closer to the commercial compounds. So one of the benefits of COAD, which is the benefits of any other large database people would be able to assemble, is that there's this hidden wealth of information within there. So we've got novel compound classes. Importantly, we've got this large set of standardized testing data, which is really set up to be employed by AI and machine learning approaches. And also importantly, it's got negative results included. But that is a bit of a problem sometimes with the AI because it's very biased in terms of 98% of our data is compounds that don't have any antimicrobial activity. And so there is this bias towards having that negative data within the data set. So I just want to finish off with a statement that came out from the WHO Director General this year. Antimicrobial resistance is outpacing advances in modern medicine, threatening the health of families worldwide. AMR is not tomorrow's problem, it's a crisis we must address today. And I think this seminar and the attendance at the seminar is indicative of the interest in trying to come up with solutions. And I'll finish there.
Yes, thank you very much, Mark, for giving us this insight. And I think, yes, you are right. So we have an overwhelming attendance today. And I think this gives us hope, yes, that people are also interested in that area. And the point that you made, standardization. So if just any researcher would include this one particular strain, for instance, or one method, so that would already help, yes. Could be a tiny add-on for each of us, but make a big difference to us as a total. So thank you so much. We the situation is that we now have immediately our second speaker, and we will have a joint discussion at the end after our 3 speakers. So it is my pleasure now to introduce Matthew Todd. Matthew is Professor and Chair of Drug Discovery at the University College London in the UK. And Matthew has a strong interest in open science and particularly how it can be used to discover new medicines. He has founded and has also several open science consortia, also in collaboration with pharma companies. Several of Matthew's efforts cover the chemical space. And he works towards also currently, I think your interests are also related to antiviral and also anti-malaria drugs, probably in addition to antibacterial. In his talk, Matthew will tackle the topic of data sharing in antibiotic research, from the early discovery to the point of translation, and he will provide insight into what we already have achieved and what data are still missing. So please, Matthew, thank you.
Great. Thank you so much for that introduction, and thank you so much for the chance to talk today. I will just try and do a very clever remote switching on of a laser pointer so you can see what I'm looking at. Okay. Hopefully you can hear me okay. I've got the twin delights of home working, of builders in the house and Thames water digging up the street right outside my house, so hopefully it's not too distracting. Okay, yes, I'd like to talk to you for a few minutes today about the importance of open data in drug discovery and what you can do with it more than your original study, so the value of it to the community. We just had a great introduction from Mark, and I am a huge fan of COAD. I think it's a transformational initiative and a really good example of the importance of open data, I think. So I guess we need to be clear about what we mean by open data. Here's some open data, which came from this lovely paper nearly 100 years ago now about penicillin. Of course, the data is there, right? And we can read all about it. This paper actually is so well known that it's posted in places where you can freely access it. And you know, there's some nice data here. There's some beautiful images, and there's some data here about the effectiveness of these molecules. You can walk to Paddington Station and see the building where Fleming made the discovery. All of this is open, but of course this isn't what we mean, is it? This is publications, and we always argue for open access publications. But what we mean by a sort of amplifying impact of the science is something else about the data and the value of the data. I guess we should then be clear about what we mean by open, and people use open a lot, and there's lots of different kinds of open. So open access is where we read papers freely, and open data is data we can use and do something with. As Heike said at the beginning, the FAIR guidelines clarify the value of data, of fair, accessible, interoperable, and reusable, and that's really where the value comes from. Open innovation is something we hear a lot about in industry, but it has quite a nebulous and varied definition depending on who's using it. And open science typically means a little bit more than all of this, where samples and licensing is clear and you can do more. And then open source, the kind of things that we do in my lab, is where all of these things are available, and anybody can participate in the project. So it's a little bit action there, a little bit like Wikipedia and Linux and things like that. And as you increase the freedoms that you give people, you just got to be really clear about what people are allowed to do, which is why Wikipedia has this license on every single page. So there's no doubt about what you can do with this freedom that you've been granted. So there's no confusion. I like to point out the value of data reuse is sometimes a bit hidden. Because sometimes the use of data sets and papers is not necessarily trumpeted because it's an old thing versus the new thing that's in the paper. But this study by the Hubble Space Telescope people is interesting and shows over time that the papers that are derived from the data that's coming from the Hubble telescope, which is in the blue there, initially were dominating, right? So the people who actually set up the studies on the telescope and got the data published the papers. And then over time, though, what happens is that the people who are using the data that's already been collected for additional papers, those papers start to outstrip the originals, and that's in the yellow up in the top of this diagram. So the data reused starts to generate more research outputs than the original data generators themselves. And I think we can see that a lot in science too, in more general science too, in biomedical research. Things like the Human Genome Project, obviously, where the original data over time, it leads to this enormous amplification of people using the data, provided it's openly available and provided the licensing is clear. So when I talk about open science in drug discovery, I mean these things, and these rules govern my lab for a couple of decades now. And we tend to point people to these rules to try and clarify exactly how things work, and they're meant to be very simple, but they have been used in these papers that are listed there as the way of working in the lab. And I guess several things have come up as useful tools to allow this, such as online electronic lab notebooks for all of the detail. So all of my students have electronic lab notebooks that are public facing and anyone can read them. We have ways of discussing what needs to be done that are also open. We favor a website called GitHub, which is very useful. And open data is the key, right? And there are ways of doing that are pretty simple in the way that we do it. We use like Google Sheets and things like Zenodo to put data in the public domain and maybe, you know, put it in places that are well archived and indexed. And then we obviously want to work with the community. So in the old days, when we started this about 20 years ago, that was very hard, but social media now takes care of that pretty well. And it's a pretty easy thing to reach out to potential new collaborators. And what we found over the last 20 years is that if you are open with your data and if you are open with what you can and can't do, including posting negatives, obviously, you tend to find that from a core, you bring people in who are interested in contributing, and those contributions are of a high quality. So we have run several series. For example, in one project that we run called Open Source Malaria, we've run several hit delete programs, and series 3 is the current most active of those, because the molecule has an unusual reaction hijacking mechanism of action, which was discovered a couple of years ago. But all of this activity and all the people who worked on these projects couldn't have done this work without the original data, which led to the discovery of these molecules. And that came from a absolutely transformational paper that came from GlaxoSmithKline Trescantos, in which the whole library at GSK was screened, a phenotypic screen against falciparum. And you know, promiscuous molecules were screened out, and the data were put in the public domain with a very liberal CC0 license. And this has kickstarted so much science, including the series 1, 2, and 3 in open source malaria. So when we talk about open data, we really must not forget to include the value of open data that comes from the private sector. And you can see from the number of citations of this amazing paper how big an impact this has had. We've extended that pattern to open source antibiotics, which is a different set of people, again, working completely openly and online. And we've had a couple of projects that have been running. One is current, which is against the Mur ligases, which are still a really interesting and valuable target in bacteria, for which there are no medicines that have been taken through clinical trials, and they're medicines that are available. And are interesting because there are several of these Mur ligases, and one molecule that can inhibit more than one could help to combat resistance, because if you hit 2 things at the same time, it's more difficult to develop resistance. And so we've been working on this a lot with core collaborators at the University of Warwick and elsewhere. And because we are very open about what we're doing, we have then enjoyed working with quite a diverse range of collaborators across several countries doing various parts of the project, who get together every month to talk about progress. Again, though, I have to point out that the most promising series, which is this chemotype that's shown in the middle of the slide here, a molecule that's highly potent against the enzyme, but it suffers from poor efflux. In other words, suffers from very good efflux, so it's poorly effective against wild type. These molecules were derived from a project run by AstraZeneca at the Bangalore site. So the data that we used to kickstart the whole project came from a parked industrial series. And I guess that's quite relevant to the whole revive idea of making sure that things aren't lost. Another series that we've looked at in open source antibiotics, again, was derived from a screen, at this time at a university, but from a group that used to be in industry, so at North Carolina, Chapel Hill. And we inherited some molecules with nice phenotypic activity, and worked with a little bit of funding across 3 universities up at the top here, and did a sort of hit to lead evaluation of some of these molecules, and got something that was nicely potent. But because we were open, we were able to leverage that to benefit from contributions from others who were not necessarily grant funded, shown in the bottom of this slide here, who chipped in because they wanted to be part of the project because it was interesting, including a private sector organization called Heike Discovery, who did some metabolic ID work for the project pro bono. And because, again, we were open, we were able to make clear the value of the compounds and the structures and the potencies and all of these things. And we were able to duplicate the impact by working with another group who evaluated the molecules against visceral leishmaniasis. And so it was a very straightforward thing to do, minimal amount of paperwork, as Mark was saying, to share compounds with another group to gain extra benefit from studying those molecules too. And I think it's really important to try to extract value from molecules that we have spent time making and studying. We've taken this a step further in the last year or so, where we've essentially pointed out what everybody knows, which is that labs are full of fridges and freezers, which are stacked full of molecules that are just sitting there, molecules that we call idler compounds because they're just not doing anything. And so we did a very simple homegrown study of just putting all of the molecules, their information and their data in a lab book, having a spreadsheet of the molecules, selecting 96 of them and shipping them out to a collaborator lab, the UK Health Security Agency, who screened them against a bunch of things, and we shared all of the data. And we've written up this paper, which is just going through the moves at Journal of Medicinal Chemistry. So the idea of showing how easy it is to archive, plate up, get molecules tested, and then share the data openly. And the value here is this sort of accidental discoveries from cross-screening. So the molecule here is the structure that you just saw from Open Source Malaria Series 3. This OSMS 106 molecule. And we knew this is a very effective antimalarial, but what we didn't know is that it had good activity against M. avium, equivalent at the moment in these preliminary studies to the potency of bedaquiline. So you know, this kind of cross-screening of serendipitous discovery is really what's on offer if you can take molecules you've already got and which are just sitting in the fridge and freezer, and if you can just get them working, you can leverage additional data from this. And I think something like this is of course baked into the DNA of COAD. So what kind of data are we missing then? Well, in an ideal world, we would have lots of possibilities for evaluating molecules in the way that COAD and the UK Health Security Agency are able to offer. But there really are very few options. And part of this IDLER project that we were doing was looking into that there really aren't that many labs that will take compounds and evaluate them and then share the data. Ideally, what you want is a simple submission process, and the data would be straightforwardly submittable to an open repository. Submitting data to things like PubChem and Campbell is not straightforward. It does take quite a bit of effort. We had a great deal of help with our submission to PubChem, which made it straightforward, but it's not straightforward. And ideally, of course, the data sets themselves will be amenable to machine learning. So one example here, which I think is a real sweet spot, will be accumulation in gram-negative bacteria. And there has been some work on this, but the more data you can generate here, the better our predictive algorithms for accumulation, I think. And it's a fairly straightforward scientific problem it's a sort of discrete problem where the measurement of these molecules, the technology for measurement of these molecules is known. Interestingly, two of the key papers from Paul Hergenrother and Helen Skurska in this area, I mean, they developed extremely interesting rules and guidelines about how compounds might accumulate. But each of them is built on actually quite a small number of compounds, about 100 in Paul's case and about 83 in Helen's case. But of course, what happens there is a small number of molecules which are available, but then the many descriptors are added on top, so the data set becomes quite large. And the importance there is to make sure that those data are shared. There's an issue there if the compounds aren't more widely available, of course, but the idea is that you share that entire set of a small number of molecules with multiple descriptors. There was some work done on this back in the day. So there was a Spark discovery platform which was made available a few years ago. This is now, as I understand it, been absorbed into COAD. So the data are made available through there. But at the time, it was surprising how little impact this had in terms of the published literature. So the paper that talked about the database itself has only been cited 8 times since 2018, which I thought was really quite surprising. It feels to me as though there is a bit of work to do here about developing a much larger machine-readable data set for accumulation, which could help solve this problem. And there are a couple of recent papers here which point out the difficulty of that, the need for open source data sets, but how difficult it is and how expensive it is to generate those in the first place. So these aren't simple problems. Reading between the lines of this recent announcement just a few days ago about the Fleming Initiative at Imperial College in London, I think and hope that developing a bigger data set that's machine readable for accumulation is part of the plan. So recent papers on the AI and generative AI highlight something I thought was quite interesting. So here are 3 examples of big, really interesting papers that are using AI in some way, including generative AI, where lots and lots of molecules are generated, and dealing with how to triage down the data to develop something which can be practically tested. So in the case of the Genentech paper up in the top left here, this involved a high throughput screen actually of about 2 million molecules, a subset of which has been released openly. So not all the molecules, because it's a proprietary data set. But the test set was enormous, and enamine was searched, so the enamine real database was searched. The code is available, obviously. And then in this paper from John Stokes, a smaller number of molecules were screened, and generative models predicted oodles of molecules, and then to be triaged by synthesizability, as Mark mentioned in his talk, so that it's useful. And again, the enamine real space was used as a sort of triaging mechanism. And then in this recent paper from James Collins, there's a big screening done, and again, generative AI models are trained on this, leading to millions of possibles, and triaged down very brutally, leading to a small number of newly discovered molecules. But I guess what I wanted to point out here is that a kind of secret source that's involved in all of this work is the enamine real database, right? A discrete set of about 40 billion molecules, which can be purchased quickly. That in itself is a very valuable commercial data set, because it means that people can find molecules that can easily be procured. And again, that shouldn't be underestimated, the value of that industrial contribution to accelerating discovery. One example I think that we could all benefit from that is really straightforward. A key component of drug discovery is solubility. We still can't predict solubility of molecules in water or buffer. Quite amazing. But if we could avoid pursuing annoying molecules that precipitate or aggregate things. So I was just wanted to point out that there's a very nice current competition which closes in January, run by people called OpenAtNet. Pat Walters is involved in this to try to help us get better at predicting solubility of molecules. So if you don't know about this, please do visit that, because there is a live challenge with real data from a real discovery program as a test set, and you're asked to predict solubility and other things of a set of molecules. So a great initiative, and I hope we see more of these kinds of things. I want to finish up with just a quick pitch to highlight something in target-based discovery. So where you're talking about trying to find molecules that may target understudied or even never studied bacterial proteins. Much of the work we are used to is, of course, phenotypic, but target-based discovery has the opportunity of maybe prioritizing new targets and new mechanisms of action which maybe we need. How then do you get better at doing that work? How do you get better at a starting point of taking a protein that you think is important for antibacterial discovery and finding a molecule that will bind it? Well, there are, of course, screening methodologies, but what we need to be able to do is predict a molecule that will bind a novel protein, and we still can't do that. So we still can't develop, a crystal structure, maybe an AlphaFold model of a new protein. We still can't predict a binder, a small molecule binder. It would be very enabling if we could do that to get drug discovery projects started. So I just wanted to highlight the Target 2035 initiative from the Structural Genomics Consortium. I lead the UCL site of the SGC, and we're involved in this program along with our partners in different SGC sites. SGC is a biomedical research charity that was founded about 20 years ago in London, and has big sites in Toronto and Frankfurt and Chapel Hill and elsewhere, and also at UCL. The idea is to go after understudied or never studied proteins and develop molecules that chemically modulate them as tool molecules that can then stimulate drug discovery. So the big aim in the next few years as part of this Target 2035 initiative, which is about finding potent small molecule binders of novel proteins, is to get much, much better at being predictive of molecules that bind. And the way that we're going to do that is to make a few thousand proteins and screen them against with a couple of technologies and release all of the data in the open. So, a few thousand proteins, a few billion molecules using techniques like affinity selection mass spectrometry and DNA encoded libraries, and then release all of the data in the public domain and work with the machine learning community to get a lot better at predicting molecules that will bind novel proteins. So there'll be a feedback loop of data and evaluation, experimental evaluation, so that the machine learners have a really big public data set to improve predictive algorithms. That's the idea. So there's a call out for people to contribute with purified recombinant proteins, for example, that people would like to have screened as part of this program. And there is a mainframe community over here for machine learners to get involved in trying to develop better machine learning algorithms for the data that's going to be coming out in the coming years. So a really exciting possibility of kickstarting drug discovery against novel bacterial targets, for example. Most of the targets will be human, but there is the possibility of doing multiple bacterial targets. If these things are seen as being genetically interesting and tractable. So I just did a pitch for the mainframe community. Please get in touch with me if you'd like to know more, or if you Google mainframe SGC, you'll find it, and it's a great community. I'd urge you to join. So just to summarize the idea here, the idea is to take many proteins, generate lots of protein ligand data, build models from the open data, make and test those predictions, and then use that to kickstart a more efficient way of leveraging that open data set, a very large open data set, to get much better at predicting starting points for drug discovery projects. Okay, last point, I just want to, because I often get questions about this. If you do all this work in the open and you're talking about small molecule binders and probes, which are potent binders that work in a cell, how can you possibly then, if everything is openly available and nothing is patented, how can you possibly then go from that to translate molecules into drugs that will benefit people? And there are many examples of this, and there's lots of different ways of doing that. One is simply to have the probe in the public domain. So up here is the potent binder of various proteins, and there are lots of other examples. And below here, you can see molecules that have been taken through clinical trials with industrial sponsors, where the molecule clearly owes a debt to the probe that was developed for it. So you can see the correspondence between these things, showing you that a probe, which is public domain tool compound, can be used as an inspiration for a medicine that is translated. And that's because, of course, there are lots of changes that you can make from a probe to turn it into a drug-like compound, which can then be subject to patent protection if that's what you wish to do. So public domainness is not an inhibition, it's more of a stimulus to private sector drug discovery. Okay, just some thanks. Yuhang Wang, sitting there in his favorite VR mask, is the guy who's working on the Malaga series, and Yinyo is the person driving the open source Malaria Series 3. I guess there's three conclusions. One is that it feels to me like open data is crucial. There's not a lot out there versus what could be out there. Two is that we have the opportunity in this day and age to improve that a lot, particularly in the AI world, but we need to ensure that the data are open for maximum research benefit. And three would be that new screening and machine learning methods bring about, I think, some fresh possibilities for target-based discovery and antibiotic research. And hopefully the SGC Target 2035 project can really help with that. Okay, that's where I'll finish. So thank you for the chance to talk.
Thank you very much, Matthew, for showing us how this can be done and giving these inspiring examples, and also, yes, new initiatives pointing out where everybody can contribute. Thank you so much. We have to move to the next speaker in the interest of time, so that we have some time for questions remaining at the end. The next speaker I would like to introduce, and I welcome, is Luiza Galarion. Luiza is a research fellow at the University of Leeds in the UK. She studied at the University of the Philippines and pursued her PhD at the University of Leeds in the group of Alex O'Neill. She stayed in this group as a research fellow, working in projects on the discovery of new antibacterial agents, also from unconventional sources. And since 2023, she is also curating the database AntibioticDB, which is supported by GARP. Today, Luiza will give us an update on new features and new information regarding this database that has been up for a few years already. and it's a very valuable resource for all of us working with antibiotics. And yeah, she will show us how we can make best use of this database. Please, thank you.
Thank you. Hopefully everyone can hear me okay. So thank you, Heike, for the introduction, and also thank you for GARDP for providing this stage to present this project, AntibioticDB, a tool for smarter discovery. So Mark and Matthew have already set the scene or the landscape on drug discovery and the importance of having relevant and meaningful open data sets. So now I'll be talking about a more specialized database. So it's been clearly shown from the earlier presentations that there are several things that we can do to curb the rise of AMR, but it remains crucial that we continue to develop new antibacterial agents, which is unfortunately easier said than done. And as you've seen, the antibacterial pipeline remains extremely sparsely populated. Right, so next slide. Right. And to give more evidence to that, last month, the World Health Organization released this report, which shows that actually only 5 drugs in the clinical pipeline from 2023 to 2025 are actually considered innovative in a sense that they don't show any cross-resistance. So we have 10 which are still inconclusive at the moment and 12 which are already showing cross-resistance. So among other things discussed in earlier presentations, I wish to emphasize this again, that one of the most fundamental challenge in sort of bringing a truly novel, innovative, new antibacterial drug candidate forward is actually identifying a suitable drug agent to develop. And it's within this context that we now present AntibioticDB. So AntibioticDB, or ADB, the link is shown here, is the first online, free, and globally accessible database of antibacterial agents. So its main purpose is to record antibacterial agents at the discovery, the preclinical, and then the clinical development, so as to provide the scientific community a repository to reassess or look at these compounds, either as a starting point for their drug discovery program, or sort of as a way to revisit discontinued or undeveloped drug candidates, especially by those intending to rehabilitate failed antibiotics to become new drug candidates again. And this point actually represents a potentially more fruitful and realistic prospect. And we have seen instances where this antibacterial compounds initially discontinued or failed to be developed or forgotten eventually became successful, such as we have daptomycin when Cubist took over its development to clinical testing, and also the development of thiacumicin B to the now approved vidaxomycin. So Antibiotic is also a resource to track antibacterial compounds of interest and a place to find compounds that show, say, for example, the same particular properties or targets. So you can look for Sabal synthesis inhibitors, protein synthesis inhibitors, etc. And when ADB was first reported, there had been no specialized database which captures compounds only exhibiting specific antibacterial activity. Right. So just to give everyone a short history of ADB. So Antibiotic database was first conceptualized and created by Laura Piddock in 2017, and that was with the support of British Society for Antimicrobial Chemotherapy. So that was from 2017 up to 2019, and at that time, Laura was also the lead curator. And over a year later, its first published report came out in Journal of Antimicrobial Chemotherapy. And before 2022, GARDP took over from BSAC, and to this day, GARDP continues to manage and support the database. From 2022, Professor Alex O'Neill from University of Leeds, together with Ursula Torretsbacher, were the lead curators of the database. And at the time as well, a team from University of Edinburgh took over all the computing and development aspects of the database. In 2023, I joined the team replacing Ursula as a co-curator for the database. And just last year, we've launched a new website here, and here we are now in 2025, where Alex and I continue to curate the database. And it's been clear how important this database is as a resource, evidenced by recent citations. And I think especially now, where more and more databases are being sought in drug discovery program. So we feel that the scientific community will continue to find this database useful as a repository, and maybe they'll find utility in projects that are looking for scaffolds to further develop into antibacterial agents here in AntibioticDB. So from its introduction in 2017, the database has massively expanded, evolved, and improved, and these improvements will be the key topics for this presentation. Right. So again, just to introduce you to the team and the key people working on AntibioticDB, we're currently being managed and supported by GuardP with Alan, Astrid, and Ali. The curation is being led by Alex and myself from University of Leeds, and all database development and computing aspects, including integration to guide to pharmacology, which I will discuss later on, is led by Jamie Davies from University of Edinburgh, Simon Harding as our developer, and James Armstrong. Right. So on the database contents, the number of the entries in the database has now actually increased over fourfold from the original 800 entries to 3,500 agents since its first publication or introduction. And previously, the key sources of antibacterial agents were publications from these journals here. But I would like to point out or emphasize these two key sources, since they have been allowed dramatic increase of historical natural product antibiotics in the AntibioticDB. And these are the Journal of Antibiotics published by Japan Science and Technology Agency and the book Encyclopedia of Antibiotics here. And thousands of these compounds have been sort of described in the scientific literature over the last 80 years or so, yet they were underrepresented in the first iteration of AntibioticDB. And it was crucial now that we included them, because most antibacterial agents in clinical use are derived from natural products, and therefore molecules of these types represent an important likely source of future treatment for bacterial infection. So apart from adding historical natural product antibiotics, the database also now includes non-traditional modalities. So what we mean by these are, those that are indirect acting, such as immunomodulators. We have antivirulence agents there as well, or adjuvants. And these non-traditional modalities were nominal in the first report of ADB. We've also now expanded to include antimicrobacterial agents, which were completely lacking in the first version of the database. Right. So to this date, this is what the composition of AntibioticDB looks like. Majority is still direct-acting small molecule antibacterial agents at over 85% here. And from these small molecule antibacterial agents, 77.5% have structure available, whereas 22.5%, it just means that we can't find any information on their structure, but are reported nonetheless to be small molecules. We have maybe around 4% which have unknown molecule types, so we don't know if they're small molecules or antimicrobial peptides and so forth. Meaning their information, structure information, is not disclosed or not available publicly. Less than 1% would be your nanoparticles and immunomodulators. Immunomodulators would include fecal microbiota therapy. We also have less than 1% for antibodies, and over 1% for bacteriophage, lysins, proteins, or bacteriophage-derived lysins, and then over 7% of antimicrobial peptides. Right. So if we look at these bacterial agents, almost 80% are still in experimental or first reports. So it's this here, meaning these are agents which are in hit to lead stages or preclinical stages, or as I said, first reports. 8% are in clinical trial here, and then another are 8% are approved drugs. 3% would have been terminated in clinical trials here, and 2.6% have been discontinued or sort of like withdrawn from the market. And these last 2 points here are quite crucial as well to capturing AntibioticDB, as the reason for an antibiotics termination or discontinuation from a market will inform researchers on what needs to be considered in their drug discovery programs. Right, so let's have a look at the web portal. When you visit antibioticdb.com, it will bring you to this home page where you will find your search bar here. In the About button, you will find a short description of the database, a video and instruction on how to use the database, and information on wildcard searching. So wildcard searching is a new addition which should help users to look for compounds of related prefix or where variation of the spelling exists. And then you can download either your search results or the full database content as a CSV file here if you wish to look at the data offline. So for a quick walkthrough, using the wildcard searching feature in the database, for example, so star here will give you the following results, and clicking on the compound will then get you to this compound landing page here. So the compound landing page would contain microbiological data at the first half of the page, and then your structural information on the second half of the page, and then finally some relevant external links at the bottom. Right. So we have now improved the information that's being captured for compound entry, and I've highlighted here the sections that are new or have been enhanced. So first, AntibioticDB has now formalized the incorporation of synonyms for compound names, and this should now improve database searching for our users. But this meant as well that it also helped us to detect and resolve duplicate entries under different names for compounds. Information on the origin of antibacterial agent is now included for more recently added entries, so they could show now whether they are natural products, and if so, if the producer organism or the name or the scientific name of the producer organism or the source. We can also put here if they're semi-synthetic derivatives of natural products or wholly synthetic compounds synthesized in the lab. And previously, this information weren't explicitly mentioned on old entries. And where available, we also put here relevant details regarding their therapeutic potential of agents. So this can be data on cytotoxicity or key results of in vivo efficacy studies. And we've also harmonized the terminology in the database with controlled vocabulary, which favored a more systematic way of managing information, such as assigning antibacterial class. So in cases where chemical compounds belong to an established antibiotic or antibacterial drug class, so say for example, beta-lactams, tetracycline macrolides, it was decided that this was the most appropriate and useful class information to display. And if that's not available, then we then put them in a more recognized grouping, such as antimicrobial peptide, antivirulence, or antibiofilm. And where categorization on these two is not possible, then entries are then categorized just according to their functional class or the pathway that they inhibit. So say, for example, if they are cell wall synthesis inhibitor or protein synthesis inhibitors. And thereafter, if that's not available, then we just put there the nature of the compound. So if it's a natural product or a synthetic small molecule. So for the new functionalities from 2019, AntibioticDB has benefited from reciprocal integration with the Guide to Pharmacology, and you will find a link to their website here. So Guide to Pharmacology is a long-established and widely used database of targets and ligands as well for both infectious and non-infectious diseases. So whereas AntibioticDB captures microbiological data and of antibacterial agents that are at any point along the discovery to the development stage, the Guide to Pharmacology actually focuses on biochemical properties and pharmacokinetics or pharmacodynamics of a ligand or of advanced antibacterial drug candidates. So what you'll find is that information in the two databases will be highly complementary. And to date, nearly 18% of the AntibioticDB entries have a reciprocal guide to pharmacology link, and we still actively collaborate with the University of Edinburgh, specifically with Jane Armstrong, to ensure the reciprocal integration is always up to date. So since the first introduction of ADB, the entries have been provided with a PubChem link where available, and now more than half of that, so 55%, have associated PubChem links. And the main aim for this was to provide users with structural information. Having said that, we have further upgraded ADB to now include structural information within the database website, which means that this will allow users to search the AntibioticDB for compounds based on specific chemical features. So this was important feature update in that it has helped us to provide structural information for compounds which do not have PubChem links, but whose structure have been represented or shown in the original report. So to date, 70% of our entries have structural information as shown in 2D structure representation, molecular weight, and the following chemical identifiers. Right, so finally, we are still doing further improvements to the database, which will include the following, so watch this space. As we now have the structural information for most entries, Simon Harding from Edinburgh is in the process of now incorporating a chemical structure search feature, which will hopefully be possible soon. And this will allow users to then search the database for similar structures or substructures using 2D representation or SMILE strings, and you would see that landing page here. Moreover, we'll be introducing additional filtering options to refine compounds based on agent type, whether they're natural product, semi-synthetic, or synthetic, by mechanism of action. So say, for example, pathway inhibitors, by activity spectrum. So you will now be able to look for antimicrobacterial and not only just anti-gram negative or anti-gram positive compounds. Or if you want to screen compounds, whether they are direct acting or indirect acting, or by a range of molecular weight. So with this, we expect to increase the quality of information captured for our entries. And lastly, there is ongoing capture of information and new entries to grow the database. And we are asking the scientific community to help us with this effort by submitting compounds or telling us if they spot any errors or want to provide additional information to this email here. And you can also see this email from our website. Right. So just to summarize everything else, AntibioticDB is a valuable data set or source of starting points for antibacterial discovery programs. And it is unique in that all entries have been reported to show specific antibacterial activity. As a repository, it has at the moment 3,500 entries, which now includes classical direct-acting antibiotics and a diverse non-canonical antibacterial agent. And as a tool, the recent addition of this chemical structure, chemical identifiers and 2D structure representation, and soon the structure searching feature expands its utility really not only to medicinal chemists, but anyone who's have interest in antibacterial drug discovery. So I'd like to finish this presentation by acknowledging our funder, GuardP. Again, the key individuals are listed here, which are overseeing the project: Alan, Astrid, and Ali. Our brilliant developers and computing and development and computing is done by a team in Edinburgh, led by Jamie. The website development is done by Simon, and the curation and integration is done by Jane. And we would also like to thank the following people listed below here, who have participated in our beta testing and have provided valuable input before we release the new website in 2024. And that's it. Thank you all for listening.
Yes, thank you very much, Lucia. It's a valuable, very valuable resource indeed, and we can only beginning to imagine how much work it is to keep it all updated. Yes, and all of your, of course, efforts, yes, and all the previous curators. Thank you so much. So we have already received a number of questions, and we have about 15 minutes for discussion time. So if you have questions, I just want to quickly remind you, this is how you can add them. There is this question button here, and then it opens up a field for you where you can type in your questions, and I can see it then. So what we will do is we go question by question, and if there are too many, so if we cannot handle them all during the time while the webinar is still online, we will try at least to get answers to you later in writing. So for the discussion session, maybe all of the speakers can come back and switch on their cameras so that people can see you when answering. And I think I directly jump into the question of our audience, and if there is some time, and if you have questions for us, yes, I'm also happy to. I'm happy to feature them too. So the first question comes from David Bickel, and it is to Mark. Mark, thank you very much for your interesting talk and the incredible valuable work on the COAD initiative. Unfortunately, the last version of the COAD dataset available for download is from 2020. Can we expect an update soon?
Yes, you can. So the first version of the database had about 100,000 compounds in it. We've got about another 130,000 compounds that are just about ready. We're just trying to get a publication out to help publicize the release of the data. So that's probably still at least 3 or 4 months away from happening, unfortunately.
Thank you. Thank you. The next question is from Fanny Violan to Matthew. Collaboration are possible outside of an open data model. What advantages do you gain from having such an open data model here? How different are your scientific activities because you are running an open model?
Yeah, I mean, we all know that, you know, more eyeballs on a problem tends to solve problems more quickly. So collaborating with people who you may not know by virtue of opening up the project can really benefit. And we've certainly seen that, where people have come to the project and done things that we couldn't. But I'd come back to this idea about if you put the data in the open, you gain extra value from downstream work, people working on the things that you've generated, which may of course come back to you and benefit. And I think that you made this point, Heike, that the funders will like that if it's open, you are trying to extract maximum value from the data and we don't lose anything. I think we all know if we're in a lab and we've got a group of students working, how much of that work sees the light of day? What, 20% or something? I don't know. It feels like by pushing things out there, you make it much more likely that useful things can be found and used by other people. So selfishly, we benefit because we get people coming to the thing that we can't do, but downstream, it's better value for money.
Okay, thank you. Yes, I think it's very reasonable. The next question goes to Lucia and Mark, both of you. Thanks for the great talks. What is the main difference or the differences between the AntibioticDB and COAD? Which one is a better starting point for machine learning models? And does the COAD database also contain data from known antibiotics? So maybe Mark and Lucia, you just maybe go on. Yeah.
Yeah. So the COAD database does not contain known antibiotics. The data in the COAD is reliant on the molecules that are being submitted by the various collaborators. So there's all sorts of different types of chemical entities in there, some of which, a large percentage of them, which don't actually have antimicrobial activity. So I guess if you're looking for a data set that also has the compounds that don't activity, COAD would be a place that has that data, whereas I'm assuming the antibiotic database, Luiza, is does not have that.
Yeah, true. So I agree. In terms probably as well on the number, when you look for training data sets, I feel like you would need more entries in there. And so far, what we have is a specialized number, and it's only like a 3,500 at the moment. And but we are looking to expand. But these are at least antibacterial agents that we know show activity. And so if you want that data, then you can get it from AntibioticDB. We also provide the current stages or the highest developmental status of the compounds that are in there too. So if you want to know more about that, you should be able to see it in AntibioticDB.
And I guess the other difference between the COAD data set, all the compounds are tested against 5 bacteria, 2 fungi under broth microdilution at CLSI-like conditions, but it's in 384 well-played assay, so we can't exactly reproduce what the recommended CLSI guidelines are. And it is against ATCC reference strains for all of those. We also have a subset of compounds that are tested against TOL-C mutant and membrane-impaired DC2 mutants and a Pseudomonas multi-efflux pump knockout, with the idea of trying to get some comparative data of wild-type strains versus mutants that have impaired efflux or import. So we also think that that data set's valuable as well. So that'll hopefully all be coming out in the next few months when we have our next database release as well.
Yeah. From my impression, I think the two databases are quite complementary to each other, right?
Yeah, I'd agree.
And yeah. And my question to Lucia, my personal question to Lucia would be, I mean, I can imagine searching and being ahead of all, on top of all the literature is a lot of work, right? So could you imagine like having a very exploratory space before being uploaded, having the entries uploaded to the database where the actual authors could be invited to fill in? Like if you reach out to the community and say, and if you are publishing a new antibiotics paper, can you please pre-fill this matrix for us so that we have just to like give it a quality mark and then it can be directly uploaded? Would that help you? So
I guess if there was a platform for us to know who we're going to send this information to or who we're going to ask this information from, then maybe that would be good. What I would say though is at the moment for authors too, so even if they've not published the compound that they're working on, at the moment, the only way for them to tell us to include it in the database is to send us the email and to say that, oh, can you include this, and provide all the information that we currently have in there. But I guess that would be really good in an ideal scenario. Then yeah, if you can just give us all, and then we can then like slowly look through it and see which we can then include. Yeah, that's great.
Yes, have the crowd help you fill in this. Yes, right. There is a question from Gabriel Kornman to Matthew. We are being asked to also share data from clinical trials. How do we ensure we are not violating participants' rights?
Oh, that's beyond my day job. So yeah, we don't do any of that kind of work, and sharing clinical trial data is a very separate question, because there you're talking about things that are being translated, and you've got to think about your mechanism of funding that, if you're going to do it openly versus you're going to do it closed, and you have all these kinds of issues to do with confidentiality. So that's not something I think that we're necessarily talking about today, and is probably a good subject for a future webinar.
More complicated, yes. Next question goes to Mark again. It's from Monica Sharma. Yes, bacteria develop evasive or resistant mechanisms against antibiotics. Yeah, what are your thoughts on the utility of bacteriostatic compounds, learning from nature's balance on colonizer and pathogenic microorganisms, along with discovery of new bactericidal compounds? I'm not sure I get this question completely. I think it's about benefit of bacteriostatic versus bactericidal agents? Yes.
There's actually been a couple of recent publications discussing the benefits or possible advantages or not of bactericidal versus bacteriostatic, and basically it's the division between the two is a bit artificial in terms of it's all based on time of exposure and concentration of exposure. So a technically bacteriostatic compound will eventually be bactericidal if you treat the bacteria long enough in many cases. And the antibiotics that are nominally bacteriostatic versus those that are bactericidal, clinically, they don't seem to see any differences in efficacy of the two classes. So I'm not sure there's a clear distinction between the two.
Right.
Anyone else, feel free to chime in, have different opinions.
One question to Matthew. You were pointing out this Target 2035 initiative and how to ensure that the bacterial penetration challenge is considered, or is it somehow incorporated to give the best chance of any identified hits also being actually able to get into the bacteria? And you know, that has been a big issue in previous screening campaigns with isolated enzymes in big pharma in 1990s, 2000s. Yes.
Yeah, absolutely. So, I mean, nothing worse than optimizing a molecule against an exciting new target and then finding the bacteria to spit it right out again. So these things are parallel projects, right? So the idea behind the going after new targets is to try to fit, well, there is an understanding biology element that you want to see the function of a protein in a cell. And one way of doing that is to pharmacologically knock it out by having a molecule that binds to it and inhibits it. It provides you greater flexibility than doing genetic knockouts. So that's one thing. So you want a molecule can actually do that. Of course, that can then be taken on to become a medicine through changes. So you want to optimize that as a tool, but absolutely you need in parallel to solve the accumulation problem. And that is not trivial. And we've all been mentioning that. But there's no way that these things that could be developed will allow you to develop a medicine unless you can solve that problem too. So these are kind of parallel things, trying to find new mechanisms of action and validating those, whilst at the same time trying to make sure that they're not useless.
Yes, thank you. I think you're completely right. We have seen various angles of how the community brain can be used. Yes, to validate targets, to finding first probes for as starting points where pharma industry can later make drops of, to have like standardized screening of a large antibacterial structural space, and also to compile all previous knowledge so that nothing gets lost in a valuable database. I think there are all different angles to the same topic, and there are many, many more things, yes, that people could do. And if we share data, it's much more fun and it's much more effective. So as I understand it, and I understood Shireen, do you want to come to us now? I think we are 1 minute short of the supposed ending time. Yes, and I would like to hand over to you for the final information. Thank you.
Sure. Thank you so much, Heike. And also thank you to everyone for your really wonderful presentations. And I also want to say thank you to the audience for your interesting questions. I think it was a great webinar and a good discussion afterwards. So thank you. I just wanted to also give a quick word about a survey that GARDP is collaborating with the European Partnership in One Health Antimicrobial Resistance. So this is a survey where we're trying to gather the gaps in strengthening the research capacity in the field of antimicrobial resistance. So if you have a few minutes, please go check it out, and it would be really great to get your feedback on that. I also wanted to give a quick word about our online conference, which is held in February 2026. So registrations for the conference are now open. Also, please check out the website, and we have a really, really exciting program for 2026. keep an eye out on the Revive activities. Some of the updates also that Luiza mentioned in her talk that will be coming out on AntibioticDB will be released in our newsletter. So feel free to stay in touch with us via there. And so, yeah, that's it from our side. Thank you so much again to everyone, and have a good day. Goodbye.
Bye.
Thank you.